Hello.
You again?
Yes, it is me, Dr. Judy Benign.
Was there a correlation between activation and that behavior? And so we, at each of those spots, we do an analysis, and then what we generally do is some kind of clustering where we say, let's throw out the individual voxels that are active on their own and just find the clusters of sort of connected voxels that are active. So in this case, we say, at each voxel, we're going to have a threshold of p less than 0.05, and then we throw out any clusters of activation that are less than 50 voxels. And this is a fairly common sort of heuristic correction that people in the literature have used. And when we do that, we get this blob. And if we pull the signal out of that blob, we see this beautiful correlation across people in activation. Turns out that those are all fake data. I just made those up. I generated random data for the behavioral data and the imaging data. and used a commonly used threshold and was able to find what looks like a really beautiful correlation between behavior and activation, and that's because we didn't exert the proper control. And these are the kind of data that you often see, or at least in the past often saw in imaging papers. Some of you may have heard about this study, which is sort of another demonstration of this, where a group from UCSB took a dead salmon, put it in a scanner, and presented it with a task, a mentalizing task, or perspective taking task. And they found that, lo and behold, if you don't do the proper statistical corrections, you can find activation in the brain of a dead salmon. And unfortunately, the press picked this up and said, oh, fMRI is worthless, where the point of the poster, I think it actually never really got published because people didn't like this paper for various reasons. But the point of the poster was, if you do the right corrections, you see nothing, coming through. But this is a sort of a heuristic correction that was often used in the literature. So we need to do the right statistical corrections. We also know that overall neuroscience, this paper by Kate Button and colleagues looked at, in particular, structural imaging and animal neuroscience studies and found that overall the level of statistical power is really low. I was able to grab some data from Sean Data to look at sample sizes and how they've changed over the last 15 years in neuroimaging. So the blue spots and the blue line are data from his meta-analysis, which is about 800 studies, which only goes up through 2010. In parallel, we took data from the Neurosynth database, which is a very large database, and we were able to estimate the sample size automatically from the abstract for about 3,000 papers. And what you see is, where we have overlap, they're fairly concordant. We get about 75, the automated technique, when we compare it to Sean's data, gets about 75% correct, so it's not perfect. But what it does is it lets us project out here to where we don't have data from Sean's manually annotated data set. And what we see is that now the median sample size for imaging studies is around 24. So what does that tell us about power? Well, we don't know exactly what the right way to compute power is for these studies. But one thing we can do is just say for the simplest possible test, for a one sample t test, let's say threshold at that same p less than 0.005 uncorrected. So for a really liberal test that we know is actually too liberal, we still see that if we compute what is the effect size we have of power to find, it's around 1, which is a monstrous effect size. Basically what this is saying, you're only powered to find huge effects. You have no power to find the more sort of moderate effects that you should be able to, that you should expect imaging data to look like. We shouldn't expect correlations between brain and behavior to have effect sizes of 1 in general because of the noise in all of the different measures. So this suggests that power in the imaging literature, even though it's gotten better, sample sizes are slowly creeping up, but nowhere near fast enough to keep up with the need for power. Another issue is the analytic flexibility that we have. So just taking one of the standard software packages, I kind of worked through a number of options and a number of kind of reasonable choices at each option. And it's easy to come up with more than 5,000 possible, plausible workflows that one could run one's data through. And then if you start looking at other packages, that even multiplies further. So we know that this can have an effect. So Josh Cart published a paper a few years ago where he took a data set, one of our data sets from a data sharing project we run, and he ran it through about 6,900 different pipelines. And what he saw was that fortunately, it was a study that my lab had conducted, and the mean results across those 6,000 basically are the same things that had come out in the paper that we reported. So that's satisfying. But what's less satisfying is the fact that you can see a range in some voxels of Z's of 6, 7, 8, huge ranges in the statistic due to differences in the analysis. And so that leads you to believe that if one wanted to, one could try enough analyses and actually find something significant even when there isn't. Finally, how many of you have written code in the last week? Okay, so a good number of us write code for our analyses. Most of us were not trained as professional software engineers and don't do the things that they do to make sure that they're not making mistakes. And we know that software is going to have a big effect. So this is, I don't know how many of you heard the story of Jeffrey Chang, who was a kind of hotshot structural biologist. He had these 3 papers in the early 2000s about the structure of this ABC transporter molecule. which turned out to be wrong. So he had to retract 3 science papers, I think a couple of PNAS papers, because of one sign change in a piece of software that his lab had gotten from someone else that completely changed the structure of the molecule. So I blogged about a similar experience that I had where I was building this classification model to try to say, can we read your mind to tell what psychological task you're doing when you're in a scanner using fMRI data? And I had people doing one of 23 different tasks, so pretty hard classification problem. And I ran the data through, and this is just my bit of Python code that I used to run it, and I got pretty good accuracy, 93% accuracy. Chance should be just a few percent, right? Because it's 1 out of 23. And I was like, if it was 100%, I would have been worried, but it wasn't. It was 93%, which said, it's pretty good.
Excuse me.
And then my postdoc, he was then a grad student, now my postdoc, Sammy Koyajo, took a look at the code, and he said, oh, you made a mistake. And it turned out that I stupidly forgot to put a little index in here. And my accuracy, it turned out I was classifying individuals rather than classifying the tasks that they were doing. And so we were still well above chance, but much, way below that 93% because of this tiny little coding error. Okay, so.
Russell, can you hear me?
Yes.
Sorry for the interruption, but I do believe that a few of us don't have access to your presentation. We don't see the screen sharing.
Oh, let me try that again.
We have your audio. We have been listening to you, but I see at least three more people after me saying that they don't have access to the presentation.
Do you see it now?
Now we do. At least I do.
Okay, great. Sorry, screen sharing.
Thank you. Sorry for the interruption.
No worries. Thanks for saying something. Okay, so what I want to do now is talk about how we fix these problems. And this is the goal of a new center that we've started. Began last April, called Stanford Center for Reproducible Neuroscience. And the goals of the center are primarily to develop a platform to help people do better data analysis. And to help them focus more on reproducibility and generalizability and less on statistical significance, which is now what people focus on when they do analyses and write papers. We also want to help people track the provenance of all of their analyses so they can go back and see not just the thing that worked, but everything that they did. And also incentivize data sharing by basically giving people free computational goodies. So this is who the center is. It's primarily centered in my lab in collaboration with Ryan Blondell's lab in the psych department. And as you'll see, we're working closely both with the Texas Advanced Computing Center and with a private company called Squishy Media, which is doing the front-end web development, and then also folks from Berkeley. I love telling the story of how this center came to be. Basically, it's from a tweet. So we had a meeting at our weekly seminar last October, and I tweeted, also discussion of reproducible research and data sharing, great job by Chris, who's a postdoc of mine. And this guy, Stuart Buck, wrote, hey, interested in chatting sometime? This is a keen interest in my foundation. So we set up a talk, and we basically looked at their foundation's website and saw how much money they gave out and said, we should come up with a moonshot. If we had millions of dollars and a few years, what would we do? So we sat down, we figured it out, we talked to them, and ultimately they funded us. So what were clearly we didn't come to this de novo. We've been thinking about these issues for a long time and have a number of different inspirations. One is a project that we started in 2009 called OpenFMRI, and the idea here is basically to share full raw fMRI data sets. And it's now come to be viewed as the main repository for the sharing of what we call task-based functional MRI. This is functional MRI when we put you in the scanner and have you do some kind of psychological test, as opposed to just putting you in and having you rest and collecting brain data. The data set is still pretty small, but it's 35 data sets, about 1,300 subjects now, which is decent, and it's being used more and more. But one of the real challenges is curation. These are complex data sets, complex metadata, and to date, all the curation has been manual. So somebody sees a data set, we have a data format that we put on the site many years ago, and we expect people to sort of put their data in it, but we never had a way to really tell them whether they'd done it right, and they never quite got it right. So the one question was, how can we sort of extend what OpenFMRI is doing to make it better and to integrate analysis with data sharing. The second is an idea that was proposed a long time ago by Steve Struther, who's now in Toronto, an idea called in pairs, which is basically he's been pushing for almost 15 years now the idea that we should move away from testing things in terms of statistical significance and instead look at generalizability. So this is basically a way of processing data that doesn't ask how significant is it, it asks to what degree are these results generalizable across different individuals in the data set in this case. We're also going to generalize it out to ask about reproducibility or generalizability across different analysis streams. Another inspiration is genomics. And any of you who come from the genomics world know that there's a number of projects there where people are basically putting tools online to allow someone to upload a data set and process it using that group's processing power. Some examples are a gene pattern from the Broad Galaxy, and the one that we're sort of most closely associated with is the iPlant Collaborative, because they're using and building the infrastructure that I'll tell you a bit about that we're going to be using. The idea of each of these is you upload your data, you want to run your assembler or whatever other processing you want to do on your genomics data, and it does it on someone else's computer and then gives you back results that you can download or visualize. And for us, the quid pro quo is going to be, well, if you use our free computing, you also have to agree to share those data at some point in the future. So what have we done so far? Well, the first thing we've done is developed a data format that we call BIDS. I'll tell you a bit about that. We're working on a data portal and on implementing analysis pipelines within that portal, and we've made some good progress there. I'll talk a bit about leveraging some tools from the software engineering world that have come to be known as DevOps tools for basically doing better science, to try to convince you that we can learn a lot from the software engineering world. I'll say a bit about power analysis and a bit about sort of addressing the social issues around replication. So first I want to show you a video that sort of highlights why we would spend a bunch of time and effort worrying about a formal data format for fMRI.
Hello.
You again?
Yes, it is me, Dr. Judy Benign. I did not think I could use your data, but when I talked to my colleague, she knew a person with a copy of the software.
Then why do you need me? Everything you need to know about the data is in the article.
I opened the data and I could not read it.
But I thought you had the program.
Yes, I have the program, but I do not understand the data.
I think you will find it as clear.
No. I noticed that you called your data fields SAM. Is that an abbreviation?
Yes, it is an abbreviation of my co-author's name. His name is Samuel Lee. We call him Sam.
I see. And what is the content of the field called SAM1?
Ah yes, SAM1 is the level of CXCR4 expression.
And what is the content of the field called SAM2?
I think that is logical if you think about it.
What is the content of the field called SAM2?
I don't remember.
What about SAM3? Is there a guide to the data anywhere?
Yes.
Great. Can I see the guide?
Of course. It is the article that is published in Science.
The article does not tell me what the field names mean. Is there any record of what these field names mean?
Yes. My co-author knows what the content of SAM2 is and SAM3. And SAM4.
Can I talk to your co-author?
Well,
I would very much like to talk to your co-author.
he was a graduate student. He went back to China 2 years ago.
Can I have his contact information?
He is in China. His name is Sam Lee.
I think I cannot use your data.
You could check the article to see if what you need is there.
Please stop talking now.
So, any of you who try to work with shared data know exactly about this experience, right? So we had, a long time ago, developed a data format that the OpenFMRI project used, but it was just developed by us, for us, without really talking to people in the community, without, kind of thinking about a broader set of use cases beyond our project. And so what we, last year, we started thinking about basically trying to build a new standard that would be broader, and that we could try to pull everyone in the field in to start using. Because if everybody's using the same format, then it makes it much easier for us to ingest the data. And so BIDS is this new format. We had a meeting here last January, bringing in a bunch of people from different software platforms to try to basically get them to agree on what needs to be in a format like this. We now have a paper under submission, and the first basically release 1.0 of the standard is now available online. So basically, we had a number of principles that we took into account in thinking about developing the standard. One is the most important thing is getting people to adopt it. So we're willing to sort of have it maybe not be perfect if we can actually get people to adopt it. Don't reinvent the wheel. Don't try to come up with something completely new. Some metadata is better than no metadata. So again, often in the informatics world, there's sort of a perfectionism about getting every possible thing one might need. We try to step back from that and say, basically, we'd rather get some metadata than none at all. We don't rely on external software like databases or on complicated formats like RDF. Everything is basically in JSON, which is an easily readable text format, or in just tabulated limited text files. And then basically, we want to capture most of the experiments that are out there, but then allow people to extend the format for data sets that don't quite fit. I'm not going to go through the details of this, but basically the idea is that part of one of the big arguments we had was, is it okay to embed semantics into the file structure, into the names of the folders and the names of the files? And that's what pretty much everybody does, right? And so we decided that that rather than sort of having complex XML files that map all these different things onto file names in a really flexible way, we basically say, here are some rules for how you name things. And we sort of came up with those to be as generalizable as possible. And then we use sort of standard formats like text files and Nifty, which is a common imaging format, JSON for sort of dictionary style data. But then we allow people to use, if there's things where nobody's developed a new format, we can allow legacy data formats as well. So we got the community involved early. So now all of the major software packages are at least working on sort of setting up their software to deal with data that are in this format. We provide a validation tool that's basically we wrote a JavaScript validator so it can run in the browser. So somebody can basically, without having to upload any data, can say, I want to see if my data follows the BIDS format. They click a button, it looks at their files, and it's actually pretty quick. It can do it in usually less than a minute, and tell you basically, these are the things you need to change in order for that to work. This file is named incorrectly, this folder is wrong, and so on. So it helps people, without us having to do anything manual, helps them get their data into this BIDS format. And then the great thing about having this format is that once we have it, we can build workflows and tools on top of it so that we can tell people, if you put your data in this format, all you have to do is click a button and you can run these analyses on your data set, rather than having to kind of work to write wrappers around your data to get code to work with them. So the idea is that we want to have adoption of the standard be a plus for people, not have it be like medicine, but have it be something that actually helps them. And then we're working with a number of different database groups to actually implement, basically being able to spit out, so when people have their data in a database, they want to spit it out in BIDS format, you can do that transparently. I won't go into all the different tools that have been developed. The main one is this BIDS validator, which is the thing that I mentioned that does the validation. But we have a bunch of other tools for both doing quality assessment on the data and doing conversion. So all of that was sort of leading up to the work that we are doing in developing a portal. So the idea is, the grand vision of the portal is somebody comes in, they have a data set they want to analyze, they click and upload the data set, they decide which processing pipelines they want to apply, they do that, they get their results, and then at some point down the road their data gets shared. Obviously that's pie in the sky right now, but I want to just give you a little tour of what it is that we're working on. So that we'll be releasing, hopefully in April, our first public beta of the platform. We have a private beta right now that does a little bit that I'll show you. But basically you can, if you just show up and you want to browse publicly, you just click on the browse publicly and you can see, oh, here's a bunch of public data sets. So if I, for example, wanted to look at this DS003, I can go say, here's some metadata about it. It ultimately will have DOIs. But we can tell you how it's licensed, and in general, all the data, unless otherwise noted, all the data are licensed by the most lenient license we can use, which is a public domain dedication, which basically says, go do anything you want with these data. And then we ask people to attribute, so basically say, if you use the data, please tell people where you got it, and also please, attribute to us that you got it from us, and share anything you do with it.
I'm sorry, Russ, can I just ask a question?
Yeah.
Why something as mild as we hope? Can't you do, you're giving them the data, can't you ask a little bit more pointedly, say the condition of use is that you
we could. The question is, what's the benefit of doing that? Obviously, it would get us, so in principle, if somebody is willing to not do this, they're probably willing to also not do that, right? And I don't want to be in the business of going and suing people for having, misused the data, or, So ultimately, I want the data to be shared and to be out there and for people to take them. And if that means that certain people will take them and not attribute to us, I'm okay with that. Some people putting data into the system may not be. For me, it's always been a trade-off of sort of ease of use and restrictiveness versus adoption. And we've heavily tilted towards getting people to adopt, getting people to use the data. And if that means we, lower the degree to which we're going to be, to get attribution for the data, I can live with that. Yeah.
Following up on that, but is this data only yours, or it comes from other studies of authors as well?
Right now, the database comes from a number of different groups, the data in the OpenFMRI database, and the first data sets to go into this project will be all the data from OpenFMRI. I think currently somewhere a little probably around a third of the data in OpenFMRI are from my lab, maybe a half. The rest are from other labs that have, at first we got an NSF grant to sort of pay for other labs to give us money. Now with the PLOS One data sharing requirement, we're getting increasing traffic of people who want to share data through OpenFMRI because they've submitted to PLOS One.
But do you expect then the authors to cite the original authors that produced the data? Do they have any plans for that?
Yeah, so we actually, this isn't filled in for this data set, but actually the idea is that we give people a how to acknowledge section here, which basically says, if you're going to use these data, this is how we would like you to acknowledge it. So if the owner of this data set wants to say, cite my original paper, or if they have a data paper, however they want it attributed, they can put that there. And then they can grab any part of the data set, the full data set, and take it and run with it. Now, if you have an account on the site, and ultimately, basically anybody will be able to get an account on the site, they'll just log in with their Google ID, and then it takes you to your data sets, and you can see you can upload a data set. So if you have a data set that's in BIDS format, you just say select folder. It will, you'll go show it the folder, and it'll allow you to give it a name. And then it looks in there. In this case, it says, we found 6 warnings in your data set. Proceed with this data set by clicking continue, or fix the issues and try again. And then it tells them, sorry, this is tiny and unreadable. This file is not part of the BIDS specification. Make sure it is included in the data set by accident, and so on. You should define effective encoding for this file. So we have a set of required metadata or recommended metadata elements, and if those aren't there, we give a warning. You can still upload the data if they're not required. There's very little that's required, there's a lot that's recommended, depending on the type of data. Then you can see the data set is uploading here, and once it's uploaded, now you can go work with it. So this is the newest part of the platform, which lets you actually, once the data are uploaded, go run an analysis on it. And if you're from the public and you show up and you see a public data set, you can run an analysis on that. If I'm logged in as myself here, actually this is our developer, and this could be a private data set that nobody else can see, and I can still run analyses on it and then decide to make it public later. But the one thing that we do is we keep track of every analysis that's been run on a particular data set, and we also require that for any analysis that's run on a data set, that data set has to be basically versioned, or we have a snapshot, so we know exactly what state the data was in when that analysis was run. Yeah.
So Russ, it says download data set there, so can't somebody download the data set and do their own analysis?
Yeah, yeah, they
That's
certainly
not
could.
tracking,
Yeah,
right?
yeah. They could either do it here or they could do it on their own machine. Yeah. And the hope is that we, by providing the kind of processing infrastructure that we're going to provide, that people will end up doing it here. But yeah, they could take the data and do whatever they want with them as well. So let's say we want to run an analysis, so we click on that button and we pick. In this case, we just have a couple of different things. So we might pick this quality assurance protocol, and then we fill in a little form that basically asks us some questions about settings for that protocol, which obviously for a more complicated protocol that could get pretty long. Then we click run analysis, it's been submitted, and then it basically will be sitting here in this thing, data set analysis status is running. Sorry, this turned out to be really tiny, but basically it just tells you what the status is of your job. And if you had a bunch of jobs, you would see them all there. And if the jobs needed to be run in a particular order, it could do that as well. So this is where we're going. This is what we hope to have sort of ready to go in April, so people can start uploading data and sort of kicking the tires on it and actually analyzing the data. One of the things that is really important for this is the engine that does all of the workflow management. And this is built on a tool that Chris Gorgolewski from my group has worked on with others for a few years now, called NiPipe, which is a Python-based workflow engine that basically has interfaces for all of the major imaging software packages, SPM, FSL, FreeSurfer. So you can basically put together a workflow, which is basically defined as sort of a graph like this, and it will, go run everything, and it's sort of aware of high-performance computing resources and so on. But it also tracks the provenance. So we have a file for everything that's run that tells us exactly what version of the software was used, when was it done, and so on. So we can deeply track the provenance. So what are we going to be able to do with these resources, or how are we going to try to encourage people to do work that's more generalizable? Well, the first is simply moving them away from looking at in-sample statistical significance to looking at generalizability. So doing things like bootstrap analysis to ask how consistent are the results across subsets of the data. Another thing that we're really interested in gets back to the analyses that I showed you from Josh Clark, and relates to this idea that John's had about vibration of effects. So the question is, if we run the data through, let's say there's 1,000 plausible workflows, how much do the results change across those workflows, and which effects change more? Which ones, quote unquote, vibrate more? We can also then start to ask questions like, we have a bunch of data, which particular workflows, we know there's those 1,000 workflows, which of them in general perform better in terms of, say, out of sample generalization? And are there data type by workflow interactions, such that some workflows work better for, let's say, data collected with a slow TR, or a slow sampling interval versus a fast sampling interval, things like that. So we can, once we have enough data, we can start to ask those kind of questions to really think about optimizing processing streams. So if somebody just shows up and says, I have this data set on schizophrenia, I want to know how their activation differs from healthy controls, we can say, here's a set of curated processing streams that we've determined to do the best job on data like yours. And obviously, if you want to try other things, you can. But the point is to sort of give people defaults that are that we know to be good. Yeah.
So how are you defining good?
Well, in terms of things like, for example, to what degree do they vary across subjects?
Things can systematically replicate and not necessarily be good.
If you put zeros in for everything, it replicates really well.
Right, yeah. Mm-hmm.
Yeah, so we could also think about having sort of detection, for example, as if we have some kind of gold standard, which we have kind of bronze standards probably. We could use those, look at our ability to detect. Now that's a really good point, that we can't, it's easy to get stuck just thinking about that reproducibility, but you also need to think about your ability to actually detect the effects are there. So we can think about simulated data, we can think about real goldish standard data that we might use to do that. But it's a J.B. Poling from Berkeley who's working with us on this. He's really interested in sort of thinking about how to address those questions.
These 1,000 workflows, what sort of parameters are you talking about here? Is this model choice, or is it?
It's generally pre-processing choice. Now, some of it's modeling, a lot of it is pre-processing. So for example, what kind of interpolation do you use when you reslice your data? what size of a spatial smoothing kernel do you use? Do you perform slice timing correction or not? And then when you get into the modeling, you can ask, what kind of basis functions do you use for your model? Do you use, this kind of temporal autocorrelation measure or that? So there's, a lot of those things. So I mentioned up front power, and power has been something that we've been interested in for a while. My former associate, Jeanette Mumford, developed some of the first power tools for fMRI. And Joka Durnes, who is a new postdoc in the lab, is now working on a new set of tools, which you can see at neuropowertools.org. And basically, what this is, it's a toolbox for doing power analysis for fMRI data. Now, it's still pretty limited. In particular, we can't quite do power analysis for the kinds of analyses that most people actually do. We can do them for region of interest analyses, which are things that some people do, but the really hard part is doing power analysis for whole brain cluster corrected types of analyses, which are what most people publish. And we have a grant submitted right now with Joka and with Jeanette Mumford to try to develop methods to do power analysis in the context of whole brain corrected analysis. And that will be the next thing that she's working on here. So, we hope that people will start thinking about this. The problem is when you do one of these power analyses, it always tells you need at least 50 subjects. And most people don't have the money because they didn't do the right power analysis before they wrote the grant, so they don't have the money to run 50 subjects. So it's really depressing when you go give talks about this stuff and you talk about these problems with power, and then people come up afterwards and say, but I only have money to do 20 subjects and I need to finish my dissertation, what do I do? And I don't know what the answer is other than, maybe go find a different dissertation, because it seems like a bad idea to keep doing terminally underpowered studies. But I think it's a real challenge. How do we adapt to the fact that half of the studies in our field at least are woefully underpowered? And clearly, part of that we can address by sort of consortia that collect larger data sets like the Human Connectome Project, but that's not the only answer. So I don't quite know what the right answer is to that problem. Okay, so I mentioned that none of us are trained most of us are not trained as software engineers, right? It turns out there's a ton of tools out in the world that people, out here in the Valley use to do better work. And we've become really interested in looking at how we can take advantage of these sorts of things. The first is virtual machines. So most of you have probably heard of virtual machines. It's basically like running another computer inside your computer. And most people in the computing world have now moved away from having their own computers. They just buy a virtual machine on Amazon, right? So the idea is you have a guest machine running on top of another machine. And the reason that we've become really interested in this is it makes it really trivial to provide somebody with everything they need to reproduce the entire analysis workflow that you use. So in particular, there are tools for what are called provisioning of virtual machines. So things like Vagrant, where you basically just have one file, this thing called a Vagrant file, that tells it how to set up a virtual machine, what software to put on, tells it where to pull your code and your data, and a few minutes later, it will have a virtual machine running on your computer here that can do all of that stuff. So we did this in the context of the My Connectome project, which was a study where I collected data over a year and a half, imaging data from myself and genomic data. So we ended up with tons of data, tons of analyses, a really complex workflow. And I started out wanting to do it in a reproducible way just because my colleagues from WashU who were doing the processing of the imaging data, a couple times they came back to me and said, oh, we've kind of changed the atlas, so here, we had to redo everything, here's the new data. So having to go and run all of these things over again without it being just a push button thing was really onerous. And so that's the first thing that drove me towards doing this, but ultimately it became clear that this is a really good way to be able to just give people your full analysis environment and let them see exactly how you did it, what you did. So we've shared this thing called My Connectome VM, which is basically just sharing a Vagrant file that you install Vagrant, you install VirtualBox, which is the software that actually runs the virtual machine, and then you just download this and say Vagrant up, and it'll pull in everything, start up with the help of Vanessa Sockett, who wrote a a lot of the sort of front end for this. It will pull up a browser to let you sort of see all the analyses. You can see exactly how we generated all the figures in the paper, and sort of browse the data in more detail if you want. It's not a panacea. There's still issues. Things can break, so it's not perfect, but it's much better than just giving out code and hoping that people can kind of put everything together and make it run. If you really want people to be able to reproduce things, it's a good step in that direction. The other thing is that we've been looking at a lot is this thing called Docker. How many of you have heard of Docker? Okay, so if you ride Caltrain, you probably hear people talking about it, the programmers on the train. So what Docker does is sort of takes the idea of a virtual machine and sort of pulls the machine out of it. So instead of having to share this, instead of having to like download an entire Linux distribution onto your machine here, you just install the Docker software, and then you get a Docker file, and it just pulls in a much smaller amount of stuff. And so this has become probably the most popular way that people sort of build applications now that can be sort of really nicely portable. And it also allows you sort of better control over exactly what the features are of the software that you're putting on, because you can control things like versions and stuff better than you can in a virtual machine. So all this stuff is coming together into what's come to be called science as a service. The idea, so in the tech world, there are all these something as a service things, software as a service, platform as a service, whatever. So people have started talking about science as a service, and the first group to really push this is a group called Agave. They're building the infrastructure for that iPlant plant genomics project I told you about. The idea is that Agave gives you a platform where basically you can describe the analyses you want to do, describe your data, and you just give those descriptions to this API. You basically push them into this system, and it will go run the stuff for you, and then sort of come back and tell you what happened. So it abstracts away from all of the stuff around, especially if you're working on big supercomputing systems. It abstracts away for the user from all of that stuff and basically just lets them describe analysis, describe data, and push it in. So what we're doing right now is working with the Texas Advanced Computing Center, which is one of the PIs on the Agave project, to basically build Docker containers for all of the different analysis packages that we do. So like the one that I showed you that was running there, that quality assurance thing, that's one Docker container that the data get pushed to, gets run, we get results back. And the cool thing about that is, we can set it up to run on one of the supercomputers in Texas, but we can also, not trivially, but not without a ton of difficulty, push it to Amazon Cloud, Google Cloud, Sherlock cluster here, maybe not Sherlock yet because it's not set up for this, but any cluster that has the Agave, is sort of plugged in with the Agave API, we can run things on. So finally, I've talked about a lot of technical stuff, right? And those of us in the room who are geeks probably think that that's like the way to solve these problems, right? But we also know, we've been in meetings with people who don't want to share data and so on, that the social parts are the really hard parts of reproducibility and data sharing. And there's, as you know from people's work in this room, there's a lot of incentives to do bad science, not that many incentives to do good science. We're starting with an initiative to create a replication award at the Organization for Human Brain Mapping. So we're going to announce it this year. The Arnold Foundation has put up money behind a prize, and so the idea will be basically, we announced it in 2016, and then in 2017, the first replication award is given to whoever does the best replication in neuroimaging for that year.
And what are the criteria for a good replication?
Well, that's a good question. I think it'll be a committee decision. And my guess is that, well, I say here, it's not even clear how to define replication, right? We don't know in imaging, what does it mean to replicate? Does it mean well, I guess I'm thinking in terms of successful replication. What does it mean for a replication attempt to be successful, right? That wouldn't be part of our criteria. I think our criteria would probably be, does it address a really important question? Does it do a really good job in terms of, reproducible practices? Those would probably be the main things that we look at. We should talk because you probably have ideas about how to do that better. It's tough because imaging studies are expensive, right? So to do an imaging study, generally, if you're gonna power it properly, so part of the problem here is going to be, if you want to do a good replication, you have to do a good power analysis, And if you want to power it properly, you're going to need 40, 50 subjects. That's tens of thousands of dollars to do that replication study. That's why people don't do a lot of replications, because they think they're boring and they're expensive, and they'd rather go do something that's going to get them a Nature Neuroscience paper rather than a paper somewhere that's not Nature Neuroscience.
I was just wondering, do you think the neuroscience community would be willing to accept, say, having the number of research projects and doubling the sample size?
No. They have to, but I think that if you went and asked, most of the people who are outside of our bubble, I think would say, hell no. You know, look, I've published all these papers, they're fine. Look, I can replicate them in my lab. You know, all the stuff that you hear all the time, right? So I think that, you know, people are going to fight tooth and nail against that.
Are sequential designs used at all?
What do you mean by sequential?
So people adding new observations and recalculating effects
Right.
and significance, and adjusting for the fact that they're
Right.
looking at each
No.
step.
In fact, the grant that I mentioned that's doing power analysis that Yoka's working on, we also propose taking some of these ideas from the clinical trial world about interim power analysis, which I assume is what you're talking about. Same sort of idea.
Yeah, I think, you know, one option is to have preset interim
Right.
analysis, but I'm thinking more of a fully sequential design. But I get a sense that most of these investigations are like, you know, you have one more subject or one more participant, so.
Right, right. Yeah, so we're definitely, I think maybe I can get you to send me some references, because I think we've been thinking about it maybe from a slightly different standpoint, but same idea, yeah. Kind of using the data you already have to figure out sort of, you know, where you can go while accounting for the fact that you're reusing the data. Yeah. So the answer is nobody's doing that yet. We hope to develop those techniques. All right. So, wow, almost 45 minutes exactly. So, you know, as you've seen, there's a lot of threats to research quality that arise from just the way that neuroimaging is done. We hope that we can develop some tools to help people do a better job. So thank all the folks who worked on this stuff, and thank you.
Could you say a little bit about debugging? So my experience, and I think sort of generalizable experience with kind of large-scale distributed workflows, is that if you immediately push data to them, they break in ways that are slow and kind of impossible to figure out.
Yeah.
So the kind of classic workflow is you do lots of intermediate checks locally on a subset of your data or something like this. You explore, and then once you're convinced that the output of a single run locally makes sense, then you push out to the cloud, because debugging in parallel tends to be difficult, and the runs are expensive and slow, meaning that the cycle of iteration is hard. How do you balance that in a web-based analytic framework?
That's a really good question. I mean, part of it is probably having, so you know, the subset of highly curated workflows that we have, I think will be, you know, written with lots of tests, right? So that we can at least know why they're failing, and so that, you know, the user doesn't have to go do that debugging. So our software engineers can do it, but they're, you know, they're better at looking into the guts of what this computer's doing. The other thing to say is that if you're, you know, these are all going to be NiPipe-based workflows. NiPipe can run on here just like it can run on the supercomputer. So one can, in principle, test it out. Obviously, there's going to be tricks moving because you never have exactly the right, you know, the same, you know, software versions and stuff. But that's, I mean, that's how we do it now. You know, we develop everything on here and then out. And Docker sort of helps with that, I think, because at least if it runs in Docker anywhere, it should run in Docker everywhere. So the bigger problem is going to be probably the interface between the Agave API and those Docker containers. See?
I want to get a better sense of the culture around sharing, what people have to lose if they share. So as you know, in the clinical research world, there's resistance to sharing because they think that they or the graduate students are going to mine the secondary endpoints and all sorts of things forever, even though whether or not they ever do. And that's part of the fear of, you know, sort of giving it away. What are the incentives? How much does that exist here? And what are the incentives besides, you mentioned one journal incentive plus one, and using your software? What are the other losses to the researchers and the gains?
So I think, clearly there's some of that fear in this world, though it's, because these data sets are generally a lot, I assume clinical trials generally cost millions of dollars to collect, right?
Some, not all.
Okay. So those issues exist here, but I think that they might not be quite as big. The one other big issue that I've seen that clearly people don't come up and say, but you can sort of read the subtext, is I'm worried somebody is going to find that I did something wrong. And we've seen that happen on a couple of occasions. And so I think that's the main fear, is that because, especially a lot of, it's easy to do imaging, right? It's easy to get somebody to help you collect some data, to go download a copy of some software and punch some buttons and get some pictures, right? Without really knowing what you're doing. And so I think particularly labs that don't have a lot of technical expertise are really worried that they're surfing right at the edge of their knowledge or beyond their knowledge, and are worried that somebody's going to find that.
So you think it's fear of finding false practice as opposed to giving away an asset of, that they want to
I think it's both of those, but I think it's, I don't know, to what degree they could both be at play, but certainly, they're both at play here. In terms of the incentives to share, one thing that I've seen is that it seems to be the case, at least amongst sort of the newest generation of researchers in this field, that there's sort of what I like to call a warm glow around sharing, that people who share data are viewed as doing better science than people who don't. And this can be gained, right? There are people who claim to be sharing lots of data who don't actually share much, but the field usually figures that out. And so I think that's a growing incentive. And also I think that when you share data, at least in some cases we've seen that people get involved in collaborations that they wouldn't have gotten involved in before. Clearly still the incentives are pretty small, and the disincentives are pretty substantial. We're trying to push against that.
I think there's also sometimes a sense that we're the experts in these particular measures or whatever, and so people don't want to share their data because they're afraid other people would use them without really understanding them and without really knowing what they're doing.
I think that's probably less the case in imaging, because it's a fairly the data are all pretty similar.
Mm-hmm.
I mean, the manipulation you did while the data were being collected is what differs.
That's
but
the
data,
imaging, but what are you correlating it with?
Exactly.
So,
Yeah. No, you're right. That people might think, oh, I'm the only person who understands this task. Yeah. Though it's funny because I would, if I'm the only person who understands the task, the alternative is, if I don't share my data, is somebody else goes and does the task and does it poorly, right? So you would think that you would want them to use your data because they would get data that were sort of well done. I mean, that's the idea behind the Human Connectome project. They've collected more than 1,000 subjects now, and they're just giving it all away, right? I mean, obviously they're doing their own analyses on it, but the point is to do a really good job of designing a study, which one can argue about whether that was fully done there, but certainly the data are really high quality, and so the idea is we give the world a bunch of high quality data, and they can go do what they want.
Nothing else? Okay, well, let's thank our speaker again.
They've done all this stuff we've never done. Yeah, it's interesting that nobody's brought that Yes, not for sharing of this kind of stuff, but actually for sharing instead of sharing of completely raw data sets, we're sharing of statistical maps and outputs of the analysis. And we've teamed with other journals to basically suggest that people upload yes, if they need to understand videos from sharing of data sets like this is a lot of procedure. Not using a format we do any other. It's a lot of part of the reason behind working with these software computers was that if everybody already has their data formatted in their then it should be relatively easy to so that would help kind of reduce the barriers. So setting a bunch of libraries and analysis and sharing this data. I mean, obviously, we're still well away from that.
And it has some weird
So we definitely, I mean, I shy away being the person who tries to get journals to require.
I'm not sure it makes any difference. It certainly doesn't take it away, and
it's pretty solid, feelings in the field. But we definitely work with journals. Because we're on the medical repository. That's as far as once we have more of a platform, we can
I mean, I was like last week, like when I shifted my feeds to say that it's probably something that sounded so plausible that they go to reader last week. And when I realized, like, you know, what they
no, that's not true. We share you. Even if I share raw, the main thing we do, we identify them facial.
No, I didn't use it. So I was like,